Good afternoon. My name is Markus Mahr from the Charité Medical Center in Berlin, Germany, and it's my pleasure to co-chair this symposium on effects of highly effective modulator therapy together with Dr. Deepika Polonini from the University of Washington in St. Louis, Missouri, and Trey LaRose, who's our community representative and will also provide his perspective on the symposium. So, as you may have noticed, the subtitle of this symposium starts with continued emerging evidence of the vicious circle on mucus obstruction, inflammation and infection. And, you know, I really think that highly effective modulator therapy has been so far the biggest breakthrough we have seen in cystic fibrosis and probably one of the biggest breakthroughs in pulmonary medicine. And we subtitled it Continued because we had a first symposium on this topic last year where we had relatively short-term data limited on adult or adolescent and adult, 12 years and older. People with cystic fibrosis, where we reviewed the data on effects on CFTR function, mucus, infection, and inflammation. And, you know, putting this together, we are really excited to have another symposium this year, again with excellent speakers, where we can feature key new developments. And these, which I think is very exciting, now include people with CF with no delta F508 mutation, but also work on effects on the homeostasis and host defense in the airways, actually down to the single cell level. Then we also have emerging data on children that probably have the largest potential for largest lifelong benefit, and then also emerging longer term data for adolescents and adults we'll hear about today. So I very much look forward to the presentations and also to the discussion, and now want to hand over to Trey.
Welcome, everybody. My name is Trey LaRose. I'm an adult with cystic fibrosis. I'm 30 years old. I've been on Trikafta for, what is it now, about 5 years, 4 years. I've seen some robust benefits. I don't no longer culture any of the long-term bugs that I had cultured for a long time, and I don't actually do really many treatments anymore. I've seen a lot of incredible benefits that has really changed the trajectory of my life, which is a pretty remarkable achievement in CF medicine. I'm really excited today because I think that one of the sort of unintended benefits of the amazing benefits we've seen of highly effective modulator therapies is that these days we're having a lot more conversations of the pathophysiology of cystic fibrosis, which I think has allowed us to start to ask a lot of really interesting questions about infection, inflammation, and the role that these modulator therapies play in treating cystic fibrosis manifestations. So I'm looking forward to some talks today, and I thank you all for being here.
Thanks to the co-chairs for a great welcome, and we'll introduce our first speaker, Dr. Pierre-Regis from the Public Assistance Hospital of Paris and University of Paris, with the first lecture, Lexicaftor, Tezacaftor, Ivacaftor effects in non-F508 del and unapproved CFTR genotypes. I have to say this is some of my most exciting topic and favorite work to listen to from Dr. Burchell.
Thank you very much for the kind introduction. Good afternoon, and I'm honored to be here today with you. And I will talk about ETI effect in non-F508 people. So these are my disclosures. Many people in the field have some disclosures with Vertex, but they have not really anything to do with what I will present you today. So we discussed about ETI, and Trey introduced us about the incredible effect of ETI, mostly for people with F508, because all the clinical trials have been performed in people with F508 variants, and thus there are people with F508 who have access to the drug or were eligible to the drug based on the labels. And, well, approved means a different thing in Europe, where I come from, and in the US. Because in Europe, thus far, it's still limited to people with an F508 variant, and people with no F508 variant have no access, as they are not eligible to the treatment. In the US, it's slightly different, and we will discuss this in depth, because there are a series of rare variants that have been approved based on in vitro data, so-called FDA-approved variants, 177 variants. Just a reminder of the worldwide prevalence of F508. If you gather data from registry around the world, you will find that approximately 82% of the people have at least one F508 variant. But there's a huge variability. In this map, the deeper the colors, the more people you have with F508. So what you see is that in Latin America, in Southern Europe, in the Middle East, in Northern Africa, you have low levels of people with F508 variant. And what this map doesn't show is that even in countries with high prevalence of F508, there is within country genetic diversity, especially in the US, where you have non-white populations who are less likely to be carriers of F508. So then we have to remember about the development of CFTR modulators and the way to go for people with non-F508 variants. These molecules were discovered by high-throughput screening and were submitted to, I would say, classical clinical development for people with gating variants and people with F508. So there were clinical trials and then proper approval by regulatory agencies, reimbursement, and use in a large number of patients. The problem is that for rare variants, is that they are rare, which means that it's difficult to make clinical trials in people with rare variants. Well, you could eventually aggregate people with rare variants that are not so rare and do a clinical trial, and this has been done. But for most people, it's impossible. So there are other ways that we will explore today. Some are cell lines, some are patient-derived cells, and some, which I like very much, are compassionate use program. So the agenda for today is as follows. We will not discuss clinical trials because there is only one trial about this, which is made by Vertex. And this trial has been published and the result has been released as press release. They have not been published, sorry, they have been presented and they are still unpublished, so I don't have any data to show to you. Then we will discuss FRAT T cells, fish or rat thyroid cells. We will discuss the interesting story of N1303K, patient-derived cells, and then the French compassionate program. So let's start with FRT cells. Well, the FRT cell model was established, and there was a publication in 2017. At that time, it was for ivacaftor. And basically, FRT cells are cells who do not express CFTR, and they can be stably transfected with mutated CFTR. And if you treat in vitro these cells, you can partially restore CFTR function. And investigators at Vertex discussed with the FDA, and they established a cutoff value at 10% of wild-type CFTR function to say that this is predictive of clinical outcome. Based on this, the FDA approved ETI for 177 rare variant in December 2020, so approximately 1 year after the initial approval for F508. One of the problems with this thinking is that this is science, but there is no really clinical data to back up this decision. And for example, in Europe, the European Medicine Agency, the EMA, didn't accept this. So because the US was one of the only countries in the world to date to have this approval, and because the US was by far the first country to get this approval, there were data in the Cystic Fibrosis Foundation patient registry about these patients. So we gather with a group of experts and we extracted the data from the CFF patient registry on people with at least one of the FDA-approved variants that started ETI between December 2020 and December 2022. And what you see here, the results have just been published in the European Respiratory Journal, and you see that there were approximately 1,800 individuals eligible to this new expanded label in the US, which represents approximately 5% of people in the US, of people with CF in the US. Of these 1,800 people, only half of them started treatment within 2 years after the expanded label. And there are reasons for that, and I don't have time to discuss it today. Interestingly, 55% of these people were already treated with a previous modulator, mostly ivacaftor. And although there were 177 variants in the label, only 92 of these variants were represented in the US, which means that many variants, not that many patients, and not all of them start treatment. So what is happening in terms of FEV1? Here you see the FEV1 percent predicted. This is in pink data prior to ETI, and in green data after ETI. You see that there's a shift. So it's an increase. This is for all subjects, and the effect is greater if you have no CFTR modulator at baseline, especially if your FEV1 was between 40 and 90%, just as in clinical trials. So if you put that in number, the magnitude of the effect may seem small. For the overall population, it's 3.3 point increase in percent predicted FEV1. But if you go for people with no CFTR modulator and an FEV1 between 40 and 90, then it's 7.5%, which is not nothing in a real-world study. BMI was unchanged in a relatively well-nourished population, but then exacerbation rates, whatever the model, basically decreased by 2 times. So it's a decrease by approximately 50% of pulmonary exacerbations. So from this, the take-home message is that at the population level, there are benefits for this expanded label for the patients who have been treated, but we have no data at the individual patient level, at the variant level. N1303K. Well, N1303K is not part of the list of the FDA-approved variants, and the reason for that is that it was tested in FRT cells by Vertex, and it was considered to be non-responsive. Then there was this seminal case report in Blue Journal in 2021. What they showed in this manuscript was that when you treat cells, so one more time, cell line, transfected with N1303K, you can show an effect in vitro. I don't go into the details. And, well, the patient is doing better. And is doing better with an N1303K and a stop codon, which means that the effect is related to the N1303K. It's doing better. FEV1 is increasing by 20%. And sweat test, well, is not that much. It was 108 before, 95 after. So after this, many groups in the world, including in France, in Israel, in the US, tried to give ETI to people with N1303K. And we summarized all the data from this group in an article earlier this year in 35 patients. And to make a very long story short, this is FEV1 before treatment. This is FEV1 after 1 month of ETI. And you see that FEV1 is increasing very much by approximately 20%. This is in all 35 patients, but it also occurred in homozygous patients, in patient compound heterozygous with stop codons, and with other mutations. So this is FEV1, but then look at sweat chloride. This is 60 millimole, the so-called cutoff for CF. And you see that even with ETI, most people stay over this cutoff, and the decrease is minimal. this was presented last year at the meeting and published very recently in the Lancet Respiratory Medicine in August 2024. This is a single-arm study from the US where they treated 20 patients with N1303K, with mostly the second variant was mostly a class 1 variant, and you see sweat chloride changes, very small under treatment, and when you stop treatment, it goes back to normal, so it's less than 5 millimole change, and then increase in percent predicted FEV1 by 10% under treatment and goes back to normal. What was also interesting in this paper was that they had models in cells, in FRT cells, and in their FRT cells with stable transfection, in-house FRT cells, the increase was plus 26%, whereas with Vertex it was less than 10%. So there is some variability, and not all FRT cells are the same. And there are also some subtleties with FRT cells according to stable or transient transfection. And here you see that in this study they had 9.5%. But overall, this shows that N1303K, which is not on the FDA list, is clearly responsive to ETI. Patient-derived cells. Well, this is, I see Felix here, and this is from his group, if I'm correct. So this is the idea that transfected cell is something, but you study only one allele in an experimental model, and it's better to work with patient cells. And the overall idea is that you would show some promising effect of the treatment in vitro, and then you would give it to the patient because in vitro data would predict the clinical outcome. This has been done with rectal organoids, and this is a study from the Utrecht group in the Netherlands. So each bar is a patient, and you see organoid swelling with forskolin, and it increases, so this is before ETI, and this is with ETI, and you can restore CFTR function in a number of people over the cutoff that has been achieved for F508. So that's interesting, but it's a lot of labor, and very few people have received treatment with this method. In France, with a group of Isabelle Samet, we studied cultured nasal epithelial cells, and one more time you can show that you restore ion transport in vitro with ETI, and a change by 5% in these primary cells was correlated with an increase by 10% in FEV1. But look at the numbers one more time, it's 39 patients, so it's something, but it's on a lower scale. So there are not many patients because this is really, one more time, labor intensive. Which leads us to the compassionate program. The idea is to bypass completely the cells and to go directly to the patients. We know that the drugs are well tolerated, that the mechanism of action is very fast, and that we have biomarkers such as FEV1 and sweat chloride. So based on this, we discussed with the French Drug Agency, and I like this one very much, especially in front of an English-speaking audience, because this is in French, so you would not understand it. But this is this is from the French Drug Agency. This is a very official document, and the translation is here. On May 19th, 2022, we got approval from the French Drug Agency to give ETI for 2 months for people 12 years and older with no F508 variants and with advanced lung disease. And after 4 to 6 weeks, we had a centralized evaluation committee who analyzed the data from the patients, clinical data, use of other therapy, FEV1, sweat test, imaging, and made a decision whether the treatment was deemed effective, ineffective, or whether we do not know. And then we need a longer period of time for evaluation, so then we would give the treatment for 2 more months. The first part of this program was successful. It was published in the European Respiratory Journal in 2023. There was only 84 patients at that time, and half of them were responders, and half of the responders had variants that were not on the FDA-approved list. Based on this, we get an extension for approval, and the extension was given on June 1st, 2023, so a little bit more than a year ago, for 6 years and older, no F508, and any level of severity. And we excluded patients basically with 2 stop codons for which we knew that the treatment would not be effective. So the results have just been published in the Lancet Respiratory Medicine last month. We treated, actually we didn't treat them. We received 500 patients, and we denied treatment to 37 who had a stop codon, and decided to treat 479. Among those, no modulator at baseline here, FDA-approved variant, 83 patients, effective in 81. No FDA-approved variant, 360, effective in 50% of them. Ivacaftor at baseline, basically effective in anyone. So what it means was that overall, 50 to 60% of the patients were responsive. They were a responder to ETI, and they had no FDA-approved variant. I'll go fast on this, but basically, this is post-hoc analysis showing that sweat chloride is decreasing in responders, not in non-responders, and that FEV1 is increasing in responders, not in non-responders. Just a few things. Some variants are rare, but they're not that rare. This is a list of variants. You can find it in the paper. So it's approximately 12 variants that represent 150 people in the program and that are all effective. And you see N1303K here is 60 patients. Based on this, we tried to derive the responsiveness of individual variants. Basically, for FDA-approved variants, it's very good. Most of them are responsive. Only those with splicing defect are non-responsive. For non-FDA-approved variants, we can use prediction from CFTR2, from other databases. And when you predict that a variant is likely to be positive, well, it's responsive. So the prediction is very good. You can use also this to predict the non-responsiveness, and usually it's also very good. But sometimes there are variants that you don't know much about. It's difficult to predict, and then it's widely spread among responders, non-responders, responsive, non-responsive variants, so you need to test. So I'm coming to an end. Approximately half of the patients were responders, and there were more responders with non-FDA approved variants as compared to FDA approved variants. And we were able to map CFTR variants, many responsive variants, many non-responsive variants, and some that we could not conclude because they were always found in responders in strands with responsive variants. So this was inconclusive. So in conclusion, there is a wide diversity in non-F508. Heterogeneity is by country, but also by ethnicity. A large proportion of non-F508 people with CF may show response to ETI. In France, it's in the range of 60%, but it may be different in different countries. It's difficult to perform clinical trials. Variants proven to be responsive to ETI in FRT cells are very likely to prove responsive in the clinic, but the opposite is not true. If it's negative in FRT cells, sometimes it's responsive in the clinic. Patient-derived cells provide very interesting information, but they are not manageable on a very large scale. And the compassionate use program provided very important data. And you can combine all this data to try to map the non-F508 CFTR variant and obtain label extension. This is a map of the prevalence of non-F508 DEL responsive variant, showing that these data have major consequences for countries in Latin America, in the Middle East, and in Southern Europe, where the dark spots show you that there is a larger number of people eligible to this variant. And this is a big thank you, and because I'm already too long, I'm ready to take questions.
Thank you, Dr. Burchell. We have 5 minutes for questions from the audience.
Hi, I'm Ian Thornell, University of Iowa. So I was wondering if you guys have looked into the cytokines in the patients who are responding in vivo, but maybe their mutation is not working in FRT cells, or maybe they only get a modest sweat chloride response. So maybe the inflammation is giving an extra expression of CFTR and bang for the buck. And if you could tease out those cytokines with samples you have.
Okay. Well, we didn't do it on a large-scale basis. This has been done in small sub-studies. Thus far, we don't have any data on that, but I would expect it would be probably comparable to what has been found in F508 patients.
Really great talk. Thanks. That's really exciting. Do you need 2 months? Did you look at some earlier time points, for example, sweat at 48 hours, and would that be discriminatory?
Okay, great question. We don't need 2 months. Most of the patients were evaluated between 4 and 6 weeks. We said 2 months because of logistic issues, so that the patient still get refilling of the treatment. Probably in many cases, the effects are more are occurring faster. So you're right, 48 hours, 1 week. Actually, when we decided to do 4 to 6 weeks, most people would say this is not long enough. You should do 12 months, 6 months, 3 months, I don't know. So this was a trade-off.
But do you have that 48-hour data so that you can go back and compare that to the longer term?
No, we don't, because this was based on a national study. So it's all the data is spread across 47 centers.
I have a completely crazy question.
I like that.
You expect that, but this is all CFTR related. But we now know that at least the lexicaftor in TRICAFTA or CAF trial is potentiating other channels like the PK channel. So when you look at your table 5 of the 2023 ERJ paper, you can actually see some patients responding extremely well in lung function with no change in sweat chloride. So how long do we treat these patients to see other effects that actually have beneficial effects on lung functions that may not be related to CFTR?
Okay, great question. Of course, I don't know the answer, but I think it goes back to what you consider as a response at the individual patient level. Is it a decrease in sweat chloride? Is it an increase in FEV1? An increase in FEV1 is something that you can achieve first by luck. Some people have bronchial hyperactivity, but also by, for example, treatment with antibiotics. So all this was taken into consideration when analyzing the files. So it was not a single biomarker, a single, effect measured. And when we didn't know, we were very conservative. We extended the treatment period. And what's happening when you do that is that CF is a progressive disease. So usually the patient, when they are not responsive, they don't go well. And after 2 months, usually the patients and the physician come back to us saying, we want to stop the treatment, we think it didn't work.
We have time for one more question.
It's actually not a question, it's a comment, and it's related to Matthias' question. Martina Gensch and I, I'm Carla Ribeiro from UNC Chapel Hill. We published several years ago studies in vitro demonstrating that if the human bronchial area epithelium primary culture, originally they were 508del homozygous, if we infect them or inflame them basically, the rescue of several modulators, including TI lately, is enhanced. Then we moved in subsequent papers to demonstrate that the mutation, the N1303K, which is not approved for FDA because the response under the regular way of doing the serotyping they're very small. But when we would inflame those cells, then they would respond drastically to ETI. And as a matter of fact, there has been a paper published by our group on the beginning of the year, and there is a paper in press now in the in the Lancet Respiratory Medicine, for which we actually do have a comment that has been accepted. And this is to say that our explanation for why, for example, in 1-303K people with CF can respond to ETI in clinical trials, but in terms of lung function, other parameters, but the sweat chloride doesn't change, is because while in the airways there is inflammation, in the sweat glands there is no inflammation. So that additional factor that would lead that mutation to respond and change the chloride concentrations in that case doesn't occur. The delta F is different because it already responds without inflammation. Of course, inflammation further enhances it. So basically the answer is inflammation. It's location, location, location. If you have inflammation and you're measuring chloride, it's going to respond.
So thank you for this comment. And since it was a comment, I can still ask the last question.
Oh, Marcus, I was going to take that co-chair's prerogative.
And this is so, I mean, Pierre-Regis, you must have thought about this a lot. So the drug was developed based on functional screening, to repair the delta F508 mutation. So now you show that it works on virtually all non-class 1 mutations. So what does that mean regarding the proposed mechanism of action?
Thank you for asking the question. What do you think? No. Well, you're right in that the correctors were identified based on high-throughput screening in F508 expressing cells. It doesn't mean that they work only on F508. So I think the concept that this is variant-specific therapy may not be right. Sorry. What you need a protein substrate.
And I'm going to take co-chair's prerogative to ask one last provocative question before we move forward, and we are keeping an eye on time for our next speaker. Based on the data that you've shown and knowing that we have increasing clinical trials coming out with potentially risk therapies for gene replacement, et cetera, in modulator-ineligible genotypes, do you believe that a potential clinical trial participant entering into one of these trials should be offered an opportunity to receive ETI if they have 2 protein-conferring mutations that are modulator-ineligible?
Fantastic question, and the answer is obviously yes.
With that, we will close this and move to our next speaker. Thank you, Dr. Burchell. It's my honor to introduce Dr. Irina Lehmann, who is with us from University of Charité Berlin. Dr. Lehmann is speaking about airway epithelial homeostasis and host defense impacts of highly effective modulator therapy, new understandings at the single cell level. Thank you, Dr. Lehmann.
Okay. Yeah, thank you very much for this kind introduction. My name is Irina Lehmann. I'm from the University Medical Hospital at the Charité in Berlin. And I'm going to talk about the perturbations on cellular level that occur as a consequence of the loss of function in the CFTR protein, and how the ETI treatment may affect the cellular dysfunctions. So first of all, I have to declare that I have nothing to disclose in relation to this presentation. Now, this thick mucus layer that is developed as a consequence of this dysfunctional CFTR channels provide a pro-inflammatory environment that is characterized by the release of inflammatory mediators, by epithelial cells, by the recruitment of macrophages and monocytes in the early phase of infection that release by themselves chemokines, recruiting further immune cells, in particular neutrophils, and that lead to a further activation of an aggressive pro-inflammatory phenotype of neutrophils. And this neutrophilic inflammation is actually the key driver of the pathogenesis of cystic fibrosis, resulting in structural lung damage and disease progression. Now, in addition to this structural damage of the lung epithelial, one have to have in mind that also the host defense is impaired by this lung epithelium characterized by a dysregulated interferon response, by reduced antimicrobial activity of the neutrophils, as well as reduced bacterial killing. And now the big question is how these CFTR modulators can reverse the situation and can finally reduce or even remove this inflammatory situation and restore the cellular homeostasis in the respiratory epithelium. Now, we all know that these ETI modulator treatment was a big breakthrough in the treatment of cystic fibrosis and now can restore and improve CFTR function and stabilize these CFTR channels. And this, of course, leads to an improvement of lung function and respiratory symptoms related to, of course, an improvement of quality of life in these patients. Now, still, whether this treatment has an impact on the basis inflammatory response in the respiratory airway and the resulting damage of these respiratory cells and the loss of function is still not clear. So there's a couple of studies showing that, in fact, the concentration of several inflammatory molecules, cytokines, and other inflammatory markers, such as neutrophil elastase activity, is reduced both in the peripheral blood as well as in sputum. And these are two of those studies, just as examples. But there's more and more evidence that even after a treatment period of 12 months, this inflammation that is that occurs in the airways, at least in adult patients, cannot fully resolve, even if those patients show totally normal lung function in response to ETI treatment. Now, we were particularly interested in the situation in this group of young patients in whom these inflammatory situations is not as long persistent as in the adults, and where we can believe or where we still think that the perturbation of the respiratory epithelial is not that strong as in the adult patients. So the ETI treatment was approved for children at 6 years and older in early 2022, and this is data from a clinical trial, phase 3, published by Markus Mall and many, many others, showing that ETI treatment in this young group of patients very, very fast and very, very strong improves lung function and leads to a very robust improvement in respiratory symptoms. Now, we were interested in monitoring the inflammatory situation in the airways in this patient group and decided to study this based on nasal swab samples because we were interested not only in immune cells and inflammatory markers, but also in epithelial cells and the interaction between immune and epithelial cells. So we decided to go for single-cell RNA sequencing and were able to study paired samples of finally 13 young patients before the initiation of this ETI treatment and 3 months after. And we also were able to include a healthy control group, which was age and sex matched. And this is data from our clinical partners, Markus Mall and Simon Graeber, showing that in those patients, very, very well, the sweat chloride concentration over the 3 months treatment period decreased, and for some of the patients, even in this to this normal level of healthy individuals. And the same was found for the improvement of lung function. Now, when you look on the cellular landscape that you can obtain based on single-cell sequencing from nasal swab samples, this is the picture you get. Actually, you can identify each and every important immune cell you will find in the airways, the neutrophil population, monocytes and macrophages, dendritic cells, B and T lymphocytes, as well as natural killer cells. Even more important, you see each and every epithelial cell you find in the respiratory epithelium of the upper airways, including these basal cells, which are the stem cells of the airway epithelium, the mucus-producing goblet and club cells, and the ciliated cells responsible for mucus clearance. And you can also identify this very, very small population of ionocytes. Now, first, we try to identify the expression of CFTR in the respiratory epithelium. And this is a so-called U-map, where every dot represents one cell. If it's gray, there's no expression of CFTR. If it's dark blue, it's a high expression. And as you may see, very, very clearly, the highest expression of CFTR can be shown in this very small population of ionocytes, but also other epithelial cells in the respiratory airway are able to express CFTR, mainly club and goblet cells, and to a smaller extent also ciliated cells. Then we went on and performed a differential expression analysis and then pathway enrichment based on this differentially expressed genes between healthy children and children before the onset of ETI treatment. And the interesting thing was that we mainly were able to show that interferon signaling is perturbed in this young group of cystic fibrosis patients, together with a perturbed regulation of viral entry into host cells. And in addition, we found several pathways focusing on antigen processing and antigen presentation. So altogether, this points to a reduced host defense in this epithelial cell layer in the upper airways. Now, when you look on the induced gene expression in this epithelial cells, it's mainly translational processes that are affected here. Now, first focusing on the interferon signaling, we defined an interferon gene set score based on all the genes that were differentially expressed in those 4 pathways that appeared here. And again, this is a U-map showing each and every cell in the epithelial cell fraction. Black if a gene is not, or this interferon gene set is not expressed. Red if there's a very high expression. And you can see very clearly that in the healthy population, we see here in particular in these club and goblet cells, a little bit also in the ciliated cells, a strong expression of interferon genes, which is completely or mainly missing in those cells before the onset of ETI treatment, but is restored after this 3 months treatment period. So interferon signaling, this is impaired in young children with cystic fibrosis, can be restored mainly in major parts by this ETI treatment. Now, in addition, as I mentioned, we saw genes affected that were related to host defense against viruses. And just to remind you, when an epithelial cell is infected to a virus, first of all, this virus is recognized in the intracellular environment by so-called virus sensing receptors or pattern recognition receptors, among them MDA5 and RIG-I. And then there's a downstream signaling that includes, for example, STAT1 and STAT2 signaling. And later, an interferon response is induced together with so-called interferon-stimulated genes. And this together defines this antiviral status of a cell, so the defense against a virus, and is called the innate immune response of an epithelial cell against a virus. Now, why I'm talking about this, we looked for different genes that are related to this viral sensing and type 1 and type 2 interferon response. And now here the gene expression is shown as so-called dot plots. So every point is the gene expression in a particular cell population. The larger a dot is, the more cells in a population express the genes. The more yellow a dot is, the stronger is the gene expression. And if you see a red circle, the difference to the healthy population is significant. And what you very easily can see that when you look on the always the left side on the healthy children, in the middle the baseline before, and then on the right side after ETI treatment, we always see a reduction in gene expression for all these genes for viral sensing and interferon response in each and every epithelial cell type, which is mainly restored, and in some cell types also overregulated by ETI treatment. So it seems that ETI, at least in this young patient population, restores the epithelial innate immune response against viruses. Now next, as I mentioned already, antigen processing and presentation was affected in epithelial cells of the upper airways. And here the same dot plots are shown, but for MH1 and MH class 2 genes. This is insofar important as nowadays we know that these epithelial cells can act as so-called non-professional antigen-presenting cells and serve as a first barrier and against invading pathogens. And in principle, you see the same picture here, and decreased expression of all of these MH class 1 and MH class 2 genes in the basal situation before ETI treatment is initiated, and a restored expression of those genes following ETI expression, or sorry, ETI treatment. Now, finally, I want to focus a little bit also on the immune cell side. The strongest biological function that we have found in cystic fibrosis was inflammatory response and cytokine-mediated signaling, as we would have expected. And again, we used an inflammatory response score derived from a hallmark inflammatory response gene set. And here again, you see these U maps with blue indicating a cell where the gene is not expressed, and red when the gene is very strong expressed. And what is very obvious here is that in particular the population of neutrophils and macrophages show this strong increase in inflammatory gene expression before starting ETI therapy, which was in major parts again reduced after initiating this ETI treatment. Now, this is a little bit another picture on the same set of genes. Here, each and every gene of those 259 genes from this inflammatory gene set is shown in this heat map. Again, blue if a gene is not expressed, red if it's highly expressed. And you can see very, very easy that in the middle, this is the expression of those genes before ETI treatment begins. The entire set of those genes is highly expressed, and then after treatment, many of those genes go back in their expression level to the normal healthy status, and half of them go back to a very low expression level, not so far away from the healthy status. So again, we can show that the strong activation of those genes that is observed in this young patient group before treatment is restored after initiating ETI treatment. And we found that in particular interleukin-1 signaling and TNF signaling was affected here and was reduced expressed after treatment. Now, at the end, I would like to show you that you can also study interactions between immune and epithelial cells, for example, by using this CellChat tool, where you calculate the interaction between the different cells by calculating the expression of ligands and receptors on the different cell types. And here the lines between the cell populations demonstrate interactions between two cell types, with the arrows indicating the cell type that is getting the signal. And here we have all the immune cells on this side and all the epithelial cells on the top. This is the situation we find in healthy children, where there is a little bit of TNF signaling between all the immune and epithelial cells, which a completely changed picture when you look in the cystic fibrosis children before ETI treatment. Here we have this additional strong TNF signal that comes from the population of macrophages, and which is mainly going to the neutrophils, activating these neutrophils that they are recruit to the airway epithelium, which is one of the most important causes that this neutrophil population is in such strong activated status. Now again, if you look after treatment, the strong activation signal coming from the monocyte macrophage population completely disappears, and this population of immune cells goes back to a less activated status. Now, just to summarize what we could show was the single cell study based on nasal swabs in this young population of cystic fibrosis patients. We could show this, that the upper airways, the upper airway respiratory epithelium of this patients is characterized by a reduced pro-inflammatory phenotype, both of macrophages and neutrophils following ATI treatment. And probably more important, we could show that the respiratory epithelium show an increased potential of pathogen recognition and interferon response, pointing to an improved host defense and barrier function, as well as a restored cellular homeostasis of these epithelial cells. Now, after only 3 months of treatment in these children, we saw that some of the cellular function went back to the level of healthy status. For some, we saw still perturbed gene expression, but this was not so far away from the healthy status. And we strongly believe that following these children for a longer time band, and we're currently collecting samples for 12 months of treatment, that this will bring those children near or already to the healthy status regarding the inflammatory phenotype and the perturbation of the respiratory epithelium. And finally, I would like to spend some words into the advantage of using these nasal swab samples for studying the inflammatory response and the homeostasis of the respiratory epithelium. These samples are non-invasive, very, very easy to obtain, and which is in particular important for children, for young children. The samples can be collected longitudinal as often as you want to collect those samples. They provide an information of both the airway immune as well as the epithelial cells and the interaction of both cell types. And what we have learned in our former studies we performed during the pandemic, that those nasal swab samples are in large parts representative for the lower, at least airway immune response. And so these are strong arguments to go for these nasal swab samples when you try to study inflammatory responses in the airways. Now, finally, I would like to mention the collaborators who were mainly involved in this study here. Our clinical partners, Markus Mal, Simon Kreber, and Miriam Feller, who provided all the samples, the clinical data, and so strongly supported the study. And in my group, Jennifer Loske from a PhD student was mainly involved in the study, and group leader Saskia Trump, and together with Søren Lucassen from the Medical Armies group. And I would like to mention that we got strong support from the German Cystic Fibrosis Association, which helped us a lot to go for this pretty interesting study. Saying this, I would like to thank you for your attention, and I'm happy to answer your questions.
Great talk, Jay Coles, Tulane. I got to ask a biased question. So you're studying the orchestration of inflammation in the nose, and you talked about the neutrophils, which I take as the violins, and then you have the macrophages, which are the horns. But the conductor is probably the CD4 T cell. So what's going on in the CD4 T cell cluster? And particularly, I'm really interested in T cells need IL-7 to survive in tissue, and now that there's so much nasal single cell data from COVID, there's actually IL-7's made by ionocytes and epithelial cells in the nose, where in the lung it's actually only made by lymphatic endothelial cells. So it's a really quite different IL-7 signaling in the nose versus the lung.
Yeah. Interestingly, there was some change also in the CD4 in the T cell population, mainly in the CD4 T cells, but it was not as strong as in the macrophages and in neutrophils, and that's why we focused on this population of more inflammatory cells. But you are right. We also could show that there were some signals coming from the CD4 positive T cells going to the neutrophils to further activate those cell population, of course.
Thank you, Professor Lehmann. I'm Clemente Brito from Yale. Our group also does immune cell profiling using single cell. We haven't been as brave to do the nose, so we use sputum. So we read your paper with great interest because we actually see similar transcriptional reprogramming after treating patients with ETI. Our work is mostly in adults, but in a large cohort of patients, we see consistent changes with what you're seeing. What we struggle with is how to separate the component that is a direct effect of ETI on the immune cells versus a component that comes from the environment change of improved mucus clearance, improved pathogen clearance. And I was wondering if you have some insight into how you would separate those, or if you see a particular transcriptional change that can help answer that question. Thank you.
Yeah, it's pretty difficult to do this in patients because, of course, the change in the CFTR channels are directly associated with the change in the mucus layer, with the reduction of pathogens, and everything goes together. And it's pretty difficult to differentiate between the different signals that are finally responsible for the status we see after ETI treatment. So I would say it's a sum of several things, improvement on several levels that finally leads to the reduction of this inflammatory potential of the immune cells in the airways. Sorry for being not able to explain this more in detail, but it's pretty difficult to do this in patients. So
Cristina Cigana from San Raffaele in Italy. Thinking to the human lung atlas and to the last studies coming from the pandemics, there is quite a different proportion of epithelial cells type through the lung, and how I think that this could impact on the transcriptomic profile in some way. How do you think that these differences can be reflected from the nasal swabs?
You mean the differences in the epithelial, in the proportion of the different types of epithelial cells? Yes. Yes, of course, this is an issue. In this case here, where we know that the damage of lung tissue and of lung epithelial cells is caused by these neutrophils. And we know that in particular the activation status of the immune cells is very, very comparable in the upper and the lower airways. We would think that the same cellular perturbation that is caused by this hyperactivated neutrophil population occurs also in the lower airways, even if the proportion of the different epithelial cells might be different, or we have additional epithelial cells present there. So it's in this case, it's not a defect that is primary on the epithelial side. It's a defect that comes that is caused by this hyperactivation of neutrophils. And so I would think it's not that relevant when you change the proportion of the epithelial cell fractions. Markus, you would?
Maybe as a comment, the main changes were observed in the secretory cells, that express, sort of as a population, the most CFTR, and they are more abundant in the lower airways than in the upper airways. So, if anything, we would work against, you would work against yourself if you study the upper airways in this context.
Thierry Ranzian from Emory. Thank you. It's a very elegant study. I had a question specifically with regards to your interferon gene score, especially if you could comment a little bit on the differentiation between type 3 interferon and type 1 interferon. And I think the increase in interferon signaling in the epithelium was kind of expected. Do you see a change in interferon signaling, both type 3 and type 1, in the immune cells as well?
Yes, we see both. Before initiating this ETI treatment, we see reduced signaling both in the type 2 interferons as well as in the type 1 and type 2 interferons. Everything, interferon gamma, interferon lambda, interferon alpha, beta, everything is reduced and is restored after ETI treatment. There's no difference between the different interferon lines.
Okay, and the immune cells also get more signaling?
Yeah, in immune cells we see in particular interferon gamma reduced, interferon gamma signaling.
Thank you.
We have time for one quick last question. Move on.
Hi, I'm Naomi from the University of British Columbia. Thank you for an excellent presentation and data. And I especially like the shout out to nasal swabs because I work with them myself. But I actually have a question that comes from that. Doing the brushings myself, I always describe it as looking sort of like a mascara brush to the patients. And so it is, non-invasive, but a little bit scrapey. And I'm curious if you think that that affects like the inflammatory state at all of the cells that you're collecting. Maybe that time course wise isn't a major concern for RNA-seq, but it's something I think about sometimes with culture studies. So I wanted to see, if you think the actual process of collecting the cells might be affecting the inflammatory state that we're seeing of them.
I would not expect that that the inflammatory status of the cells is affected by the process of collecting. What we see is that the proportion of the different subtypes, how many epithelial cells, how many immune cells you collect, which type of epithelial cells, whether you collect squamous cells when you have very deep, when you collect very deep swabs, this will, of course, be affected by the type of sample collection, yes.
Thank you, Dr. Lehman. Thank you to our speakers for...
Thank you. I'm excited to introduce Dr. Felix Ratchen from University of Toronto. His talk is titled, What's Old is New Again, Emerging Evidence on Mucus Obstruction, Infection, and Inflammation in Children Taking Highly Effective Modulator Therapy. Please welcome Dr. Felix Ratchen.
Thank you. Thanks for having me and inviting me to the symposium. I think this is a very interesting topic. As we move to children and younger children with this treatment, because as Markus has already pointed out, the younger we go, the more likely we will see a reversal of changes that have happened. And then the question that comes back is how effective is this treatment? Is it actually going to be enough in terms of controlling CFTR function in the long run? So I can't answer all these questions, but I will try to show you some of the data that have been accumulated over time. And of course, I'm going to cite Marcus Moll again, like the other people that have done this before. So I have some relationships with Vertex, but they are not related directly to this talk, but I'm talking about ETI. So I will review the current evidence about mucus obstruction in children with CF on ETI, and there are still lots of things that we do not understand. And also I want to focus on to appreciate how ATI affects airway infection in children, and I'm only going to talk about children because Dave in his next talk is going to talk about adults, and I will have some data from the PROMISE study, but I promise to limit it to the children. And then I will also, we have had quite a discussion already whether ETI controls inflammation in children, and I think there are still some questions to be answered. So this is kind of the old CF pathophysiology that we depicted a couple of years ago before we had highly effective modulator therapies, with the mucus plugging being the major effect that we see in the airways and the downstream effects of defective mucociliary clearance, changes in the airway pH related to mucus secretion and infection and inflammation. And if we know from the data that we have for ETI to date is that it affects multiple organs, but what we have seen mostly and what we have studied best more than other aspects is really the effects on the airways, and we know that it has quite significant effects on airway disease, both in the upper and lower airways. So some of the other components are probably relevant as well, but less well understood, and I'm going to focus mainly on the airways in my subsequent talk. And one of the questions that we really grapple with is how effective is highly effective modulator therapy? It certainly has been a major game changer in cystic fibrosis, but does it control all of the downstream effects in a way that we don't have to worry about them anymore? And that also is important for us as clinicians in terms of all the treatments that we have asked our patients to do. And so we don't have the answers to these questions, but if we look at some of the open label extensions of the randomized controlled trials, it looks like everything is wonderful, right? So the FE1 improvement is substantial, and it continues over time, and there's no decline in lung function whatsoever. So if that is true, we could basically sit back and say, okay, we might be done, and we at least are those patients that respond to ETI, and as Pierre-Regis has shown this, the number is substantially larger than what we studied initially. We should be in pretty good shape. But this is why it's important that we do observational studies, because the observational studies give us more of a real-world experience of what's happening in patients being treated. And so I will show some of the data from a number of observational studies, starting out with the mucus obstruction, for which we actually have the least evidence yet, and then moving on to infection and inflammation. This is from the RECOVER study that is happening in Ireland and parts of the UK, mainly the Brompton. And these are some imaging studies using CT as the most established imaging technology today. And this is at baseline and after 12 months of treatment. And you can see that trapped air is going down. It doesn't seem to be going away. But the mucus plugging, based on their data, is largely resolved after 12 months of treatment. And certainly a number of CT studies show that is one of the effects that you can see relatively consistently in patients that are being treated. But is the mucus really normal? And here's some data, again, from the German study that Marcus Moll has been leading, and that's been published in the ERJ in 2023. And you can see, if you look at different aspects of mucus property, and these are patients that are 12 and older, so not the young age group, there is certainly an effect on different aspects of mucus rheology. On the left-hand side, you see the elastic module, in the middle, the viscous modulus, and to the right, the mesh pore size. And all of these things improve towards normal. But it is very important that there's also a healthy control group in this study, because you can see that even after 12 months of treatment, things are better, but they are not normal. And so, it has already been highlighted that we need longer-term observations to really clarify whether this trend over time is going to continue, or whether there's a residual component of mucus abnormality. Because if that's the case, then that also raises the question whether the other downstream of that, downstream effects of CFTR dysfunction will still be there over time. And it also affects how we should treat our patients. And you all know about the Simplify study, that in the short term, if you stop Donase alpha or hypertonic saline in patients 12 years or older, it doesn't seem to have a short-term effect on their pulmonary function. What we're really interested in, of course, is what's the long-term impact of that. And so if we have such an effective CFTR modulator which improves mucus clearance, and we have seen this, what does that mean in terms of all of these other therapies? But there are some data that will give you a bit of a different view of what lung function looks like over time. And these are data that will be presented tomorrow by George Solomon in workshop 22, and looks at the trajectories of FE1 post-ETI in 3 different groups of patients. So these are 2 pediatric groups, part of the PROMISE study, the pediatric PROMISE cohort, and then the adolescent PROMISE group and the adult PROMISE group. And the way these data are shown is that you see that the lung function slope before patients were treated with ETI, and then there's this increase, and then you see what's happening in the year afterwards. And interestingly enough, there's actually a decline in lung function in the pediatric groups post-ETI. And that certainly is interesting and maybe a little bit unexpected from what we've seen from these open label studies. So whether this is caused by patients changing their treatments, and we've learned that happens quite a bit, that patients stop their other therapies, is still a bit of an open question. It certainly raises that question, and Scott Sagal actually will show some of these data in terms of how treatment changes after introduction of ETI. And you can see those data on poster 831. And this is summarizing data from the PROMISE study for chronic therapies, and just showing the number of chronic therapies. And what you see is over time, and this is a total of 42 months, and this is, there's an increase in the percent of patients that do not do any of these therapies, and a reduction in all of these components of CF therapy. So as we move along and think about these, how ETI affects lung disease, it certainly will be important to better understand whether what we see is just a change in treatment behavior, or whether it's related to the level of effectiveness of CFTR modulators. And just a few more data from the PEDS PROMISE study, and this is I will focus on the microbiology data mostly. So just to give you a bit an overview of this is a sub-study of the overall PROMISE study for patients that are 6 to 12 years before they go on ETI. And the primary outcome measure for that was actually the lung clearance index, as a more sensitive measure of lung function in patients with more preserved lung function. And secondary outcome measures are the typical suspects like FE1, sweat chloride, CFQR, and so on. And just to give you an idea of the population, this is a bit of a busy slide. But if you look at the FEV1 predicted at baseline, it's actually 107%, so quite good, and that is that shows how well physicians and care teams take care of their patients even before ETI. And if we look at the level of abnormality for lung function, you can see that only about half of them had abnormal lung function, even if you base it on LCI. So this is the pattern of response for the main outcomes. So LCI went down, not quite to the level of what we've seen in the interventional study, but this is a healthier group. The FEV1 response was a little bit interesting in a way that there was some increase even at the 3 months time point, and then a decrease over time. And CFQR improved, as we have seen in other studies. But let's focus on the airway cultures. And one of the problems with the airway cultures that we have in these young patients is that we mainly have to rely on throat swabs because they don't produce sputum. But we have a subgroup of data for sputum, and I'm thankful to Pradeep Singh and his group for providing these data, and I'm going to show them to you in a minute. So these are the throat swabs data, and you can see for the main two bacteria that we see in cystic fibrosis, Pseudomonas and Staph aureus, there's barely any Pseudomonas at baseline in this group of patients, and therefore not much of a change over time. If we look at Staph aureus, I wouldn't focus so much on the qualitative culture results because these semi-quantitative assessments are not very reliable. But the percentage of patients that are negative for Staph aureus goes up a little bit, but it doesn't change dramatically. So what does it look like for sputum, which we can see as a bit more of a reliable measure? So let's first look at total bacterial burden, and this is work that was done by Sarah Morgan in Pradeep Singh's lab. And you can see, and I here showed on the left-hand side the pediatric cohort, on the right-hand side the adult cohort. And you can see that there's basically no change in the total sputum bacterial burden. And that's actually similar to what has been seen in adults. But if you look at Staph aureus as the main pathogen that we see in this age group, you can see that the sputum density decreases in subjects that are positive, but it mostly doesn't go away. And what this reduced density of Staph aureus means in terms of downstream effects is still a little bit unclear. And if you look at overall what you find in these samples, really the dominant bacterial DNA in sputum comes from all organisms. And that speaks a little bit to the quality of the sputum as well. But certainly the relative proportion of bacteria that we consider being pathogenic is relatively small. And actually, if we look at changes for all of these, the only thing that really changes is Staphylococcus. So, and just to finish up on that, in those that have no low Staphylococcus, there's really no change in what you see in these samples over time. Okay, so how about the inflammation? And we already heard about inflammation and the single cell data, so I didn't want to mention any of the single cell data because they have already been a focus of a previous talk. But this is again from the German study looking at the typical marker neutrophil elastase activity in sputum. And again, you can see it goes down over time and with a little bit of wobble over time. But even at the 12 months time period, even though it looks like it's almost at the baseline of healthy controls, it is not entirely normal. And that's also true for the membrane-bound neutrophil activity, which shows a similar pattern. So what else? What other data do we have available? So Scott Sagel in a workshop tomorrow will show some data from the PROMISE study related to both serum markers of inflammation and sputum markers of inflammation. So on the left-hand side, you see sputum neutrophil elastase at baseline, and then up to 24 to 30 months, so a longer time period. And there seems to be an overall trend of this going down. But again, it doesn't really go down to zero in terms of the sputum neutrophil elastase activity. On the right-hand side, you see high sensitivity CRP as one marker of inflammation, of systemic inflammation. And again, you can see this pattern, and this is over a bit of a shorter time period of it going down to a lower level. And again, it will be interesting to follow that over time, because I got some data from a study from Canada. This is from the Adult Center in Vancouver, where they looked at this over a 2-year period. And if you look on the left-hand side, the CRP levels certainly go down at the 6 months and 1 year time period. But after 2 years, it actually tends to creep up a little bit more. And so the question is there going to be persistence of this inflammation or even rebound? We'll have to see that in longer-term studies. So that actually brings me to the end of my talk. People with CF on ETI certainly have less mucus plugging, but data suggests that mucus abnormality do not completely normalize. But we really need longer-term studies to clarify that, because if that's the case, then the question is how much is tackling mucus and addressing mucus as one of the problems with therapeutic interventions really going to be important. And that's certainly something we struggle with in our pediatric patients in terms of how much airway clearance and mucoactive treatment they should receive. The bacterial density decreases especially for Staph aureus in pediatric patients. But it's also the case that Staph aureus doesn't seem to be eliminated from the airways. And we had the discussions for decades how we should treat Staph aureus in CF patients. But I assume this discussion will continue in terms of how we best address that. Inflammation improves, but so far data suggests that it does not normalize. And if an infection persists, it would also not be surprising that this does not normalize. But really what we need to know, what are the functional consequences in the long run, because so we need to continue to follow these patients in these observational studies for longer time periods to really know what's going on and to also to guide our treatment decisions in the long run. And with that, I'm happy to take some questions.
Hello, is it on? Yeah, on?
Just shout.
Really important data, thank you for showing that. So seeing the staph sort of persistence, and there's a number of other papers that are showing this as well, do you think that's because of its more natural reservoir in people in the upper airway? And do you think that the future of this disease will continue a fight with staph?
Yeah, I mean, if you take the data for populations, you would expect that 25 to 30% have staph in their upper airway. But if you look at the numbers in CF, they're much, much higher. So they're more in the 60, 70% range. And so I think the sputum data are probably more reliable to say that those are really in the lower airways as well. So I think my bias is that the data would suggest that staph is an ongoing issue in CF. But again, it's also the question, is there a level of density where these bacteria are less relevant? And is this lower density really at a point where it doesn't require the aggressive treatment that we have thought about in the past? And that's still an unknown.
Really enjoyed it. Staph persists, and Pseudomonas was very low at the beginning, and it kept very low. So does acquisition of Pseudomonas decrease?
That's $1 million question. So there's some data now that would suggest that there's some registry data that are coming out that suggest that yes, the rate of Pseudomonas acquisition is lower. It's certainly not zero. We still see Pseudomonas acquisition in patients on ETI. So, but it also raises the question of whether patients would be able to spontaneously clear those Pseudomonas, right? Because in infants, there's this nice study from the Netherlands that I like to quote, where they studied healthy kids over time, and they developed Pseudomonas during viral infection as well, but cleared this spontaneously. So the question whether we need to be as aggressive of treating it is really a difficult one. But since the treatment is so successful, I'm not sure that I can convince clinicians to do that study.
Is there a correlation between the drop in respiratory function and the increase in inflammation and bacterial burden during the long term? And is there a correlation with the adherence to therapy, or is it possible to understand this?
Yeah, these are very important questions. So I think we would like to see clear and simple correlations between changes in lung function and inflammation. And I actually asked that question this morning for one of the talks. But it's so far the data don't suggest that there's a very close correlation. There's an overall group link, and in terms of what was your second question? Oh, adherence. Yeah, I wish we knew how to monitor adherence. We have no idea what our patients are doing. That's really the problem, and so it's. I know Paul is standing up because he has some data related to that. But it is very difficult to capture, and probably the best way to capture adherence for at least some of the inhaled medication is to have a device that measures that directly. But most of the longitudinal studies have not included that, so we don't know. John.
Thanks, Felix. Great data. Professor Lehmann was super optimistic that the improvement in the inflammatory profile was going to continue to improve towards normal. But that was not what was coming out here.
We need to be a bit controversial, right?
Yeah, well, I guess, can you hypothesize perhaps as to why that plateau seems to be, is it a plateau, I guess, first of all, or is it continued improvement?
Yeah.
Slower improvement. And is it immune reprogramming that maybe if you get in earlier, you would see more improvement? Is it persistence of pathogens? Is it maybe that CFTR needs to be even better corrected, or something else?
Yes, those are all important questions, and I wish we could answer them. I think I'm not quite that optimistic, and I know that I've also liked to study the nasal epithelium, but there are some limits to it, right? So in terms of the more lower level of inflammation and all these questions of how this is related to structural lung disease and lack thereof, and whether if we actually can have this population of patients that are relatively pristine in terms of their structural lung disease and follow those over time, whether you can completely downregulate inflammation in those. I would think that's more likely to be the case, and I think it's pretty unlikely to be the case in those patients that have structural lung disease, because we have non-CF bronchiectasis and they have quite a bit of inflammation, right? So and they have decline in lung function and all of that. So I think this is where we need also to link this to the imaging data to really clarify that over time in these populations, and just using functional data will not be sufficient for that. Paul.
Just to not leave that question on the adherence unanswered, we have some data we'll present tomorrow at the Lungs and Beyond session to answer that question, but it's really, really vital. We don't do a good enough job on it, and we need to get better at it, because that's going to be the next frontier, I think, after we improve everything, we have a whole generation of kids who are much healthier. Adherence is going to be the big problem, so we need to pay attention to it, I think, and there's some interesting data we'll present tomorrow from Recover.
Mm-hmm. Yes, sure, come on. Oh, I shouldn't say anything.
So just as another follow-up to this, how much do you think that this potential resurgence of inflammation is impacted by the de-escalation of the other therapies that you covered?
Well, that could well be the case, and I think we need to study that because that could be one of the issues. I mean, we're in the planning phase of a study of how to deal with airway clearance therapies in children, and I think those are some of the things that we need to look at because that's the only way we will figure this out.
Felix, so along these lines, what do you think will the relative role of, I mean, as Jonathan already said, improving CFTR function further, towards normal, versus symptomatic therapies, because this will not be enough? What do you think the relative role will be in the future, in the pediatric population?
Yeah, I once had to write a chapter about CF in the future, and I don't want to read it anymore because probably everything I said was wrong. It's very hard to predict, but that's exactly why we need these studies and why we need these data, because we can dissect some of that, because otherwise we will always wonder, right? So because then 10 years from now we will still be sitting there, oh, is it that? Is it this? So we need to clarify that.
We still need to leave time for Dave to predict the future, so we'll let our
okay, sorry.
Jonathan, we've got disciplined speakers on time and robust questions, so Jonathan, last question.
Sorry, very quickly. The LCI dropped and stayed flat, and the FEV1 did this sort of little boomerang thingy. I'll leave you to comment on that, and then I'll...
Yeah, so the LCI data are not suggesting worsening over time. I just used the FEV1 data to make a point here. So, and actually in our data that we're showing on a poster here, LCI continues to improve for over a 1 year period. So there will be some differences between the different cohorts, and in the end, we probably have to throw together all the data to really make sure that we have the numbers to really reduce the variability of all these signals, and then clarify what's noise and what is signal.
Thank you to our disciplined speakers and our robust audience questions.
Thank you. And I'm happy to introduce Dr. Dave Nichols of the CF Foundation, who's going to speak on long-term data on inflammation and infection in adults. I'm looking forward to this one.
I'll return the favor. Maybe as one of the simplified PIs in defense of Trey, I think it also behooves us to ask if we need symptomatic therapies, are the existing ones the right ones? So that's a bit more complicated. Thanks very much to the moderators for the invitation, and it's nice to see you all. I was asked to address the topic of long-term outcomes with infection and inflammation, particularly in adults with CF, and my disclosures are there, no personal disclosures, just an employment disclosure. This is really my outline slide, just to describe to you all how I've approached this topic. First, in regards to long-term, I've tried to limit the data to those in which we have ETI use at 12 months or longer. In regards to infection and inflammation, really for the sake of time, I've restricted this to studies coming out reporting sinus disease and airways disease. And then in regards to highly effective modulator therapy, this has the prior talks has been restricted to ETI, where the application is probably the largest. And as I was certain would happen, the adult cohorts are those greater than 12, so you'll see a little bit of overlap with some of Felix's data. And 20 minutes goes fast, so I'm going to go fast. The red light's been flashing the whole time. Let's start with what we're learning about sinus disease in regards to infection and inflammation. So the first paper I want to highlight is this one, coming out from a group here in the U.S. where endoscopic examination of the sinuses was done to use swabs to obtain sinonasal samples from an adult population, looking before and then about 9 months into ETI use. And not surprisingly, before ETI, the microbiota diversity and richness were both lower than a healthy population. That was particularly true for those who had had surgery, altered architecture in the sinuses. This didn't dramatically improve or really change much at least 9 months into therapy, and the total bacterial density, as has been highlighted before, didn't really change based on 16S technology with ETI, but Pseudomonas did decrease, the core pathogen in this group, both by 16S and then quantitative PCR work, as you can see there in the figure on the right. It's also occurred in a couple of patients in the Burkholderia genus in that population. The next study I want to highlight is a similar approach, endoscopic examination of the sinuses, and this time obtaining maxillary sinus washes in a group of about 20 individuals with pre-existing sinus disease, looking at about 10 months into ETI use. In this group, we're really focused on inflammation and signaling related to inflammation, including metals. I've highlighted just a handful of the changes they observed in this paper, but important ones like IL-8, IL-22, and lactate were all substantially better after ETI use in these sinus samples. And then lastly, work from Dan Beswick and colleagues going out now 2 years into ETI. They were focused more on clinically relevant outcomes, including symptoms, but they did imaging in this, and so I'm using sinus opacification as a surrogate for mucosal inflammation here. And you can see in the representative figure there, improvement by 6 months into ETI use and really further improvement in this individual. So overall, they saw that these improvements they saw pretty quickly after ETI use in the sinuses were stable over time. That was actually despite decreasing use, the theme for the day, of some of their other non-modulator therapies focused on sinus disease. It's also important to note, also has been highlighted that this did not reach normal. True also for the sinus symptomatology as measured by the SNOT-22 score. So what are we starting to learn about sinus disease? I think it's great. We're seeing studies focused on sinus pathology. It is a major morbidity for our patient population. Total bacterial density in these samples appears to be not largely changed, but key and core pathogens do seem to be significantly improved, and really within the first year of use of ETI. And along with that, we see associated improvements in sinus mucosal inflammation, along with structural improvements, at least as measured by opacification, likely representing underlying mucosal swelling. Let's shift now to the airways. And the first study I want to note here is, so we went back, sorry. Apologies. First study I want to note is work that was briefly highlighted earlier as well, a paper coming out last year from Ireland. Within this, there were a group of 10 adults with CF who were able to provide sputum not only before, but then 12 months into their use of ETI. You can see that this was a fairly advanced pulmonary disease population based on their FEV1, and the majority of them were chronically infected with Pseudomonas. They focused on sputum and then serum markers of inflammation, and I want to highlight something I think was really cool in this group. They recruited a matched non-CF bronchiectasis cohort of similar size and similar FEV1 at baseline. You can see their underlying demographics were somewhat different. So I don't expect you to read all of this, but I just want to note that sputum inflammatory markers all decreased through 12 months into their ETI use in this population. And they approached or really came very much in line with the levels that they measured in their non-CF bronchiectasis population. I think that's really an interesting finding. And you can see that that's true of important neutrophilic inflammatory signaling as shown in the top, as well as IL-8 and IL-1 beta on the bottom. In this group, the majority had Pseudomonas aeruginosa, and in that subpopulation, so to speak, in those folks, this did improve, though didn't really resolve in these adults with CF. And along with this, they saw improvements in serum inflammatory markers. These data were shown also. And again, we're seeing levels that by 12 months reach, or in fact, sometimes are a little bit better than is seen in the non-CF bronchiectasis population, suggesting perhaps an increased overlap in these two groups of individuals. The next paper I'll highlight, as everyone else, great work out of Berlin that came out in the ERJ last year. And so within this, again, there was a group studied about 30 individuals with CF who provided spontaneous sputum samples through a year into ETI use. And here the comparison wasn't non-CF bronchiectasis, but rather healthy volunteers, allowing us a different way to kind of consider these analyses. So the first thing I want to point out here on the left is that in these sputum samples, there was really little change in the total bacterial load in the sputum. It's notable to me that this did reach the value of total bacterial load measured in the healthy volunteer population in the far left at 12 months, but it didn't really change with ETI use. On the right, you can see that sputum alpha diversity, evenness, and richness did improve. Not all of that, but at least a portion of that appeared to be due to decreasing Pseudomonas dominance over time, as shown in the red portion of those figures. This group also went on to look at sputum measures of airways inflammation, and you can see, as I've highlighted, the healthy control population on the left, and then the 12-month data there on the right. Improvements in what was seen reminiscent of what was seen in the work out of Ireland. Important improvements, though not full normalization, compared with the healthy population and key inflammatory outcomes. I'll then move to the PROMISE data, where these data have been previously shared at prior meetings, reporting the 6-month data on airways infection. I want to call out my partner in crime, Pradeep Singh, whose lab has spent a great deal of effort and should be credited for a lot of important data provided to our community here. And along with that, the follow-up to this has just been accepted in the same journal. So the 3.5-year data on microbiology, you're welcome to see that. It's not very pretty as a prepress, but we promise to improve it. So we're talking about 175 folks who provided sputum to us either spontaneously or through induction before and then subsequently after starting ETI, now out to 4.5 years. So that's the data we can share. Thanks to, again, Sarah Morgan and Pradeep for providing these. So we are including the final study visit, although perhaps it won't be the final study visit, I don't know, but 4.5 years into ETI use in this adolescent and adult population. Just to orient you, the red line represents the cohort average, including everyone, including those who were culture negative at that time point. The cohort is defined by those who were infected with Pseudomonas before starting ETI. The purple shows those who remain culture positive, and that distinction I think is just important to appreciate, in that those who don't fully clear their infection, because we don't often get quantitative culture data in our clinical practice, they continue to have substantial Pseudomonas burden, though it appears relatively stable. So as a group, it's very encouraging to see a perhaps 3-log reduction in Pseudomonas in this population, but those who remain infected continue to have concentrations that are relatively high. The other point to make here is that we don't see any convincing evidence of a rebound effect, and that is important now that we follow these out to 4 or 5 years, because other data have suggested that that's a possibility, and we know that may still be a possibility. It may occur later, but thus far we haven't seen convincing evidence of that. I do want to highlight that the final study visit is incomplete, and so we may come back with a different story next year. If we think about Staph aureus, it's a very similar picture, where fairly rapidly within the first several months of ETI use, we have a big drop in the density overall, but then that's been stable over time through now 4.5 years. And this is all despite, as has also been highlighted, decreasing self-reported use of chronic pulmonary therapies by this cohort of individuals providing the sputum samples. You can see, for example, chronic inhaled antibiotics started at about 60% of the cohort, and it's now down to maybe 25%. That's a substantial change. As was seen even at 6 months, this phenomenon has continued through now 3 or 4 years into follow-up of the population, wherein those who came in the study before their ETI with a lower burden of Pseudomonas were the ones who were able to consistently and repeatedly shift to culture negativity, and may have eliminated at least most of their Pseudomonas in their lower airways. And so you can see that that occurred actually for the majority of those in the lowest tertile of Pseudomonas density, and none of those in the highest tertile of Pseudomonas density. So this has been, thus far, our strongest predictor to say who may be able to go on to convert to a sustained and repeated culture negative status. But there's a caveat there, because if careful ddPCR methods are applied to these culture negative samples taken even months after individuals or years after individuals have converted to culture negative, you can still identify at least small amounts of Staph aureus and Pseudomonas in the majority of those culture negative samples. Because of the size of the cohort, we can comment, at least preliminarily, on some of the less common, but yes, very, very much problematic pathogens that are difficult to treat at times. So you can see here for Stenotrophomonas, Achromobacter, Burkholderia, we actually have seen a similar pattern. Stenotrophomonas, for whatever reason, has been more likely to convert to culture negative than some of the others we've monitored. But we've seen decreases in density for Burkholderia and Achromobacter as well. But to turn the coin over a bit, we do see some new positive cultures. So here we relied on registry data to look back and identify groups who did not grow Staph aureus, Pseudomonas, or Stenotrophomonas for at least 2 years based on their clinically obtained cultures before starting ETI, and then asked how many of them grew that during our observational trial now out several years. And so the N is the potential group who, for example, didn't have Staph for 2 years and then have provided us sputum samples. There would be 22. The gray, or the higher percentage, are those who have grown that pathogen once in the PROMISE study, and the black bar are those who have grown it more than once. And so in some ways, it's encouraging to see that actually only 1 out of 67 individuals in the PROMISE study who didn't have any history of Pseudomonas for 2 years prior has now repeatedly grown Pseudomonas during PROMISE. I don't know if that suggests decreased susceptibility over time or not, but I think that's generally an encouraging finding thus far. And those who do grow perhaps new acquisition infections are doing so at a relatively low density, suggesting that, again, they may be able to clear these, and that may be why we're not seeing so many individuals with repeated or consistently positive cultures apart from Staph aureus, as you can see. This was noted before, which was terrific. I'd just like to reiterate and encourage everyone to see Scott Sagel's presentation in Workshop 25, where he'll share, really for the first time, the inflammatory data from the PROMISE cohort. It's the same cohort that was used for the microdata. And so down the road, we'll be able to ask these questions about connections or associations between infection and inflammation in this group. But both in the airway, based on sputum neutrophil elastase, and the blood with serum high sensitivity CRP, we see improvements that really mirror the microbiology changes, with substantial decreases in inflammation within the first months, and then general stability thus far, as has been measured. The last piece of data I'll just highlight out of the PROMISE work is this report that was published earlier this year, led by Sarah Morgan out of Pradeep's lab. So Rob Kramer at Dartmouth and Pradeep had developed a PCR methodology to measure Aspergillus fumigatus in sputum samples directly, because fungal culture was not part of the PROMISE study to begin with. It wasn't built in. And so we devised a way that we could assess, at least at some level, the Aspergillus burden in this population. And so the first thing I'm showing you are the registry data of the cohort of interest here. If you look backwards before ETI, 2 or even 4 years, about a third of their cultures were positive for Aspergillus, a group that was at least intermittently positive for Aspergillus. I want to note that these are likely not people struggling with conditions like ABPA. Chronic use of oral steroids was not allowed as an enrollment criteria, and so that's not who we're talking about here. It's more of the colonization or intermittent colonization with Aspergillus group. Using the PCR methods, very similar to what was seen with the bacteriology. Within a month of ETI initiation, the participants that were positive for Aspergillus fumigatus substantially decreased, and that's now been maintained encouragingly through a year or two into therapy. So I think the story about Aspergillus and among ETI users is very much unwritten or incomplete, but these are, I think, on the encouraging side of the story. going to now shift a little bit to the last set of studies I want to describe for you, which is really nice work, again, out of Dr. Singh's lab and colleagues, where they have extremely carefully gone down and collected airway samples. And they did this with single-use bronchoscopes. So we're talking about 5 different bronchoscopes per individual at 2 different time points. And they did this because they were very interested in minimizing as much as possible any contamination or cross-contamination regionally within the lungs. And I won't go into the details, but hopefully many of you were able to see Sam Durfee present this work earlier. If not, there will be posters in the exhibit room. The first thing I want to point out is, unlike all the data we've really seen from samples collected either from the upper airway or through the upper airway, and by that I mean the sinus samples I described first, or sputum samples, including those from PROMISE, where we've not seen a change in overall bacterial density using 16S and ddPCR, there was a substantial decrease in the bacterial density when measured directly in the airway, and I find that to be interesting. And likewise, as has been seen with sputum, we see substantial decreases in Pseudomonas density, measured both by culture and ddPCR. Again, each of these dots represents one of the segments of the lung, so there would be 5 per individual, and then at each of 2 time points. And the same thing is seen in staph. Very impressive decreases overall. And along with that, as we might hope, we're seeing substantial improvements in the inflammation measured directly in the lower airways. Here on the left, you have IL-1 beta and IL-8. This is a log scale. These are highly significant and important decreases in airways inflammation, though they are not returning to 0 or normal. On the right, perhaps the most intriguing data, at least to me here, is that middle figure. So neutrophil elastase, long thought to be a driver of progression of airways disease in people with CF and neutrophilic inflammation. In those segmental airways where Pseudomonas was cleared, that any level turned to 0. It was not detectable in all but one of those segmental airways. And that was not seen in those segmental airways where Pseudomonas continued to be found, where any activity was measurable and identified. And so at 18 months into ETI use in this group of adults, there was a very tight association between any level and PA culture density with an R-squared value of greater than 0.75. So I'll stop by summarizing 5 key takeaways, at least in my view, of what's out there in literature in regards to this topic. First of all, we do see a lower burden of traditional pathogenic bacteria. That's evident in sputum samples, airway lavage samples, and now in early reports from sinus samples. Secondly, the total bacterial density in sputum and sinus samples appears largely unchanged, but at least in one important study, it does appear to decrease in samples collected directly from the lower airway. Thirdly, this repeated and sustained sputum conversion to culture negative status for Pseudomonas is occurring, but we think we have to be very careful. It's occurring far less than prevalence data from registries around the globe would suggest. And those registries have been very careful to identify important caveats in the data, but I just don't think any of us should be misled. This is happening less, and if we look very carefully, especially with PCR methods, it's happening fairly rarely. Fourth, some of the less common pathogens where we have less opportunity to study these conditions also seem to be improving, and that may extend to Aspergillus fumigatus, at least in those who are more colonized rather than struggling with bronchitis or ABPA. And lastly, and very importantly, I think neutrophilic and related inflammation is improving along with microbiology, and that's true now in airway lavage fluid, sputum, as well as serum markers. It remains above healthy controls, but it does seem actually very similar to what is seen in non-CF bronchiectasis, at least in small studies, and I'd love to see more work like that. So I'll stop with that. I want to really thank all the participants, investigators, staff, and sponsors who've made all these studies possible. A big shout out to my co-investigators in PROMISE, especially PROMISE PI, Dr. Singh and his lab. Unpublished data that has been shared and the funding for some of the work I was involved in. Thanks very much.
20 minutes goes fast indeed. We have time for questions for Dave.
I'm going to kick us off. I have one question. What do what does the bacterial density of exacerbations look like? Does that look different?
I think the reports have been conflicting. There are reports suggesting that bacterial density or infection may increase during exacerbation. There are others that don't find that consistently, and I'm not personally aware of studies post-ETI that have looked at that question.
Quick follow-up is I'm going to go ahead and speculate that there's probably a lot of low-level exacerbations that the doctors in this room are probably not knowing about because patients are probably not rushing to call quite like they used to. I can speak from personal experience.
Thanks, Trey. Yes.
Hi, so Dave, thank you very much. A bit of a curveball. So you showed that there's improvement in sinus disease with ETI. So the question comes up in the lung transplant population, who a certain segment had serious sinus disease that really doesn't respond to anything.
Yeah.
There's been some small case series. Is there anything more we're trying to get that approved in Canada?
Yeah. It's a great point. Just in case anyone didn't hear, because it is an important point, sinus disease continues to plague people with CF after a lung transplant. It may be an important source of infections that ultimately affect the transplanted lung. The question is, what work is being done there? I can't commit to say that I know. I've heard discussions probably similar to what you all are undergoing in Canada. The challenge, in part, of the numbers of transplants happening now, and those who would be eligible for ETI. So I don't know if there are active studies that have been funded, but I know that there are proposals underway. If anyone in the room does know, please correct me there and let us know.
Hi, Luis Faberio from Orlando. Have you looked into mycobacteria?
We have not looked into mycobacteria in this population. We would, yeah, we have. The answer is we have not looked into it in this population. There may be opportunity through some indirect detection methodologies to do that, but NTM culture was not performed.
Thank you, Dave. Can you comment a bit on people converting from sputum producers to sputum non-producers? Because I think that's something that I worry about in pediatrics all the time. What are we missing? Especially the Reeves data about the neutrophil elastase being so closely correlated with Pseudomonas. If we're missing Pseudomonas because they're not producing sputum, it just makes me nervous. And how do you think that could have affected the results here?
Yeah. It's not a situation of what you don't know can't hurt you, right? It's a concern. We did see, despite not heroic, but substantial efforts to collect sputum in this cohort, in particular, a diminishing ability to do so even by induction. So we were down to, I think, around 50% success toward the later points in the trial. That might argue that these results could be underestimating the benefits, because those who become unable to provide sputum even by induction, may be those less likely to have at least high density infection. But that's all conjecture, and that's the problem, right? We don't know. There is a lot of parallel work to develop non-sputum reliant detection methodologies and some cool advances in that way, and maybe that'll have to be our approach.
I might just jump in on that. Some data that I shared with you, but you couldn't include on. So in Recover, we specifically started collecting nasal lavage because we knew this was going to be a big problem. We haven't published the data yet, but we've a lot of numbers now over recurrent or repeated time points, and it's very similar. Very, reduction in some inflammatory markers, but not all. But it's not that dramatic, and there's still a lot of inflammation there, and that's in both sputum producers and non-sputum producers. So I think you're right, we need a better surrogate, and it's one of many. But I think it's the same message. There's still stuff there.
Yeah. Thank you, Paul. Yeah.
Dave, I have a question. Are you also looking at viral infections in PROMISE? And the background of the question is related to the data Irina showed, that interferon signaling seems to be impaired in CF at baseline and is restored with ETI. So the question is, could that then clinically, with the next virus hitting the patient, could that then lead to, viral exacerbations we have not seen before?
Yeah. The short answer is no. That there may be opportunity, again, very indirectly using biobanked serum or plasma samples to try to get at some of this question, but that would be very incomplete, I think. We debated and we did not collect exacerbation data in this study for logistical reasons and burden on participants. We have some regret. But we did not do that, and so we don't have not only viral-induced illness, but any exacerbation data.
We'll have a final question from Namsu.
Okay, Namsu Ju from Stanford University. My question is not limited to your talk, but it's an excellent talk. All the speakers. The question is whether there is any relationship between CF patient with chronic beta-adrenergic drug user and efficacy of HEMT, such as ETI. There is any negative or positive relationship between those?
I'm sorry, the door closed as you were mid-sentence. Could you repeat it?
My question is whether there is any relationship between CF patient with beta-adrenergic drug, chronic beta-adrenergic drug users and efficacy of HEMT, such as ETI.
Yeah. So is there a relationship between beta-adrenergic drug use and HEMT efficacy? And I'm aware of interesting and important preclinical data supporting that potential interaction. We have very few users of those drugs, and we didn't collect detailed medication use. We focused on 4 chronic pulmonary therapies plus azithromycin. And so I can't answer that from this cohort. Perhaps others can comment, but yeah, they say unfortunately not. Thank you.
Thanks. Thanks, Dave. This is our second time as your co-chairs going rogue and foregoing panel to allow all the robust Q&A. So we just want to thank all of our speakers and the audience for a fantastic session. Thank you.